Assalamu alaikum طلاب. Today we will start our first lectures of concerning the histology of the organs associated with the digestive systems. So we have the liver, gallbladder and pancreas, and today we will start with the liver. The objectives of today's lecture. By the end of this lecture you will be able to describe the histological structure of the liver with a special emphasis on classic hepatic lobule, hepatocytes and their surfaces, portal tracts, hepatic blood sinusoid, space of dizzy, bile canaliculi, and we will know much more about the functional units of the liver. As you know, the liver is the largest internal organ. You can see here the liver in this image. It's the largest internal organ, weighing about 1.5 kilogram or forming about 2% of the total body weight. It's located in the right upper quadrant of your abdomen, that's to say, an area called the right hypochondrium, and it is covered with a thin capsule that send connective tissue septa to the inside of the organ, dividing it into loops and lobules. Liver has wide range of functions, many functions. Maybe the most important of them is the production of bile, a complex substance that is important for the uptake of fat from the small intestine. However, it has an important function in the synthesis and secretion of plasma protein, detoxification of large number of toxins that can enter into our body through the digestive tract, protein metabolism, carbohydrate metabolism, and fat metabolism all took place within the liver, and finally storage of a large number of molecules, mainly vitamin A, vitamin D, and vitamin B12 and other minerals. To have a much better understanding about the histology of the liver, we will start with the vasculature of the liver. We should know much about the vasculature of the liver. So the liver receives blood from two sources, as it is clear here. The liver receives blood from two arteries. 75% of the blood entering the liver came through the portal venous blood, which carry blood from the digestive tract to the liver, and 25% of the blood supply came through the hepatic artery. The hepatic artery carry the oxygenated blood to the liver. However, the portal vein carry deoxygenated blood. However, this blood is rich in nutrients and rich in metabolites. These two blood supply, these two sources enter into the parenchyma of the liver, and they fuse together, forming a network of anastomosing sinusoids. As you can see, the terminal branches of the portal veins and the hepatic arteriole run together in the connective tissue surrounding the hepatic lobule. These branches run in the connective tissue surrounding the hepatic lobule, together with a branch from the bile duct. They form a tract called portal tract or portal tract. Within the hepatic lobule, the blood from these two sources mix together, forming a common network of anastomosing sinusoids. So we have a branch from the hepatic artery that fuse with a branch of the portal vein, sinusoidal hepatic sinusoid. This is a better image here that illustrates the vasculature of the liver. We have the portal tract here, a branch of the portal vein, a branch of hepatic artery, and a bile duct. The branches from the portal vein and the hepatic artery, they mix together, forming a sinusoidal channel called hepatic sinusoid. Blood will flow directly from the periphery of the lobule toward the center. Blood will flow from the periphery toward the center, where it joins the central terminal venule. This means that the cells receiving the more concentration of oxygen are those in the periphery. The cells receiving the more concentration of oxygen are those in the periphery of the lobule when we compare them with those in the center of the lobule. But at the same time, they receive the highest concentration of other substances absorbed from the intestine and coming through the hepatic vein, such as amino acids, metabolites, toxins, etc. This difference in concentration of oxygen and metabolite reaching the liver cells has a big effect on the properties and function of these cells. So the liver cells, or called hepatocytes, near the portal areas, here in the periphery of the lobule, or we can see them here, hepatocytes here in the periphery of the lobule, can rely on an aerobic metabolism, and usually they are more active in protein synthesis, while the more central hepatocytes surrounding the central vein, they receive to lower oxygen concentration and lower nutrient concentration. So they are more involved with detoxification and glycogen metabolism mainly. Then the hepatic sinusoid. This is a picture of a hepatic sinusoid. This is the space where we have a branch of the hepatic portal vein and a branch of hepatic artery and a branch of bile duct. This area called a portal tract or portal tract. And as we said previously, it is located in the periphery of hepatic lobule and the connective tissue septa. Branches from the hepatic artery and the branches of hepatic vein fuse together, forming a hepatic sinusoid. Hepatic sinusoid. The blood will flow from the periphery, from these two blood sources, toward the center of the hepatic lobule, toward the central vein. And as we said, these hepatocytes in the periphery of the lobule receives the highest concentration of oxygen as well as the highest concentration of materials and toxins, while these here receive the lowest concentration of oxygen and the nutrients. So let's have a few words about the hepatic sinusoid. This is a schematic diagram of the hepatic sinusoid where we can see this is the space where the blood flow. This is the lumen of the sinusoid, and here are the hepatic cells, or called hepatocytes. The hepatic sinusoids are highly specialized capillaries. These sinusoids are lined with thin, discontinuous, highly fenestrated endothelium. As we can see here, the fenestrations within the endothelium of the sinusoid. The sinusoids are surrounded on all its surfaces. We can see here, this is a sinusoid. The sinusoid is surrounded by large number of liver cells or hepatocytes. And due to these fenestrations on the wall of the sinusoid and this discontinuity of the basal lamina, the plasma will escape from the sinusoid to a narrow space between the sinusoidal wall and the hepatocyte. This space is called the space of Dizzy, or called the perisinusoidal space. Within this space, many microvilli project from the hepatocytes into this space, maximizing the surface area of hepatic cells that are exposed to blood or that are in contact with the blood. So we have large surface area for uptake or release of materials between the hepatocytes and the blood. In addition to the endothelial cells, we have other types of cells that are functionally important found within the sinusoids of the liver, within the hepatic sinusoid. First is Kupffer cell. Kupffer cell, we can see it here. First is Kupffer cell. These are specialized macrophages found within the sinusoid lining to recognize and phagocyte bacteria, debris, even aged erythrocytes that are present in the portal blood. So these are Kupffer cell with its immune function. We have another type of cell that is stellate cell, hepatic stellate cell. They have small lipid droplets within its cytoplasm. And their main function is to store vitamin A and other fat-soluble vitamin. And also they are important because they produce the extracellular matrix components and cytokines that help regulated Kupffer cell activity, the Kupffer cell activity. Okay, so is it clear that we have the hepatic sinusoid? Here we have the hepatic sinusoid filled with RBCs, okay, surrounded by large number of hepatic cells, hepatocytes, and between the wall of the hepatic sinusoid and the liver cells, we have a narrow space called the space of Dizzy. This is a photo taken from the histologyguide.com, the atlas, the virtual atlas that we rely on studying histology. As we can see, this is only a part of a hepatic lobule. These are plates or cords of hepatocytes. Between them, we have sinusoidal space, and this slide actually we have ink injected into the liver of this animal. This ink is taken up by the macrophages, and that's why we can very clearly see Kupffer cells within the liver parenchyma in this slide. This is the end of part 1 of this lecture, and we have the other part of the lecture where we will discuss the hepatocytes and hepatic lobule and the functional arrangement of the liver. So see you in part 2, and thanks.